My name is Alana Jackson, and this past summer I had the privilege of working in the lab with Dr. David Granville with instruction from Dr. Steve Shin on a project entitled Granzyme B in Acute and Chronic Wound Healing. Before I delve into the specifics of my research, I'd first like to provide you with a bit of background information to further your understanding of my topic. Wound healing and tissue repair is the primary focus of many projects in the Granville lab. The wound healing process is very complex and occurs in 4 overlapping stages, beginning with hemostasis. Hemostasis is the process in which blood flow to the wounded area is stopped with platelets and fibrin. Hemostasis begins right after the wound occurs and lasts until the bleeding stops. The next stage is the inflammatory stage. Immune system cells such as neutrophils and macrophages fight potential infections at the site of injury. The inflammatory stage begins shortly after the wound occurs and lasts for approximately 3 days. Proliferation is the process in which keratinocytes and endothelial cells migrate to the wound. It can take up to 10 days for a wounded area to become fully re-epithelialized. Myofibroblasts also help speed wound repair by contracting the edges of the wound. And lastly, the remodeling stage occurs. Fibroblasts create the collagen extracellular network and to restore skin to its undamaged state. The remodeling stage can last 4 years. The difference between a wound either healing or developing into a chronic wound is a very sensitive balance. In order for a wound to achieve closure and to heal, a combination of inflammation to prevent immune system complications, angiogenesis to reestablish circulatory system connectivity, cellular activities to close the wound, and proteolytic activity are necessary. This figure illustrates the situation that occurs when this balance is disturbed. Chronic wounds exhibit chronic inflammation, impaired angiogenesis, impaired cellular activities, and excessive proteolytic activity. Granzyme B is a granule-secreted enzyme, hence the word granzyme. There are 5 granzymes expressed in humans, but granzyme B was of particular interest to our lab. Granzyme B is a serine protease, a biological scissor, if you will. And in its traditional intracellular role, granzyme B is secreted by natural killer and cytotoxic T cells, and CD8 positive T cells to induce apoptosis in virally infected or tumor cells. Granzyme-mediated apoptosis depends largely upon perforin, a protein that disrupts the cell membrane, allowing granzyme B to enter an infected cell. However, recent research has revealed that granzyme B has several extracellular roles as well. Contrary to previous belief, granzyme B is expressed by cells other than natural killer and CD8 T cells. Mast cells, activated macrophages, basophils, neutrophils, and other cell types secrete perforin-independent granzyme B and just release it into the extracellular matrix. Perforin-independent granzyme B has several capabilities, but primarily it cleaves major components of the extracellular matrix, such as decorin, a protein essential in collagen formation, and fibronectin, a protein essential in keratinocyte migration. In addition, extracellular granzyme B cleaves and processes cytokine IL-1 alpha, exacerbating inflammation or encouraging chronic inflammation. A previous study investigated the effects of granzyme B on wound healing. Apolipoprotein E is a glycoprotein that possesses immunosuppressive roles in addition to its ability in lipid transport. APOE knockout mice are susceptible to chronic inflammatory skin diseases and serves as the chronic wound healing model in this experiment. According to the 2013 study, granzyme B presence was significantly upregulated in the wounds of APOE knockout mice compared to wild-type mice. Excessive granzyme B cleaves several key extracellular matrix proteins and led to impaired wound healing. When granzyme B is not present, healing is greatly improved, leading to the conclusion that granzyme B plays a detrimental role in the healing of chronic wounds. In addition, the Granville Lab also investigated the effects of Granzyme B using a chronic diabetic wound model instead of an APOE knockout mice one. The results were similar. This research involved known Granzyme B inhibitor, serpinacrein, and revealed that serpinacrein accelerates tissue repair in a chronic diabetic wound. So from this, we can conclude that Granzyme B is detrimental to the healing of chronic wounds, but its implications in an acute wound situation remain elusive. So this information led us to form the hypothesis that although high levels of Granzyme B in chronic wounds delays wound repair, Granzyme B may have positive implications in acute wounds. There's got to be a reason why it exists. This figure depicts the wound healing model used in mice. A 10mm punch biopsy is taken from the posterior region of the animal, and the wound healing process is monitored closely, with pictures taken every single day up to day 16, when a wound is expected to be fully healed. In addition, a full histological analysis is performed on days 2, 8, and 16 to closely monitor each stage of the wound healing process, from inflammation to proliferation to remodeling. Using this model, we observed the healing process in wild-type mice and Granzyme B single knockout mice. In an assessment of wound contraction, Granzyme B presence actually increased the speed of contraction in wild-type mice, particularly at days 10 and 12 with statistical significance. Despite the accelerated healing process exhibited by the wild-type mice in addition to the Granzyme B knockouts, it's important to note that all wounds achieve closure by day 16. Next, I quantified the rate of re-epithelialization and identified the scab fall-off date for each experimental group. The wild-type mice experienced re-epithelialization at a faster rate than the Granzyme B knockouts. As depicted in the figure to the left, on day 8, the wild-type wound is completely re-epithelialized, whereas the granulation tissue in the Granzyme B knockout wound still has quite a distance to migrate before achieving wound closure. In addition to a faster rate of re-epithelialization, the scabs on the wounds of Granzyme B knockout mice, in wild-type mice rather, fell off almost 3 days before the scabs on the wounds of Granzyme B knockout mice. In order to assess the maturation of the healed wound, I quantified both the epidermal and granulation tissue thickness of each population at day 16. As a healing wound matures, the epidermal thickness of the new tissue gradually returns to the thickness of normal skin. As shown in the figure to the right, the epidermal thickness of each population is essentially the same for normal unwounded skin. However, at day 16 in the wound, the epidermal thickness of the Granzyme B knockout is much thicker than the wild type, so it's in a less advanced stage of remodeling. The same results were observed in the assessment of granulation tissue thickness. Once again, the wild type mice are in a more advanced stage of remodeling. Another indication of wound maturation is levels of collagen present in the granulation tissue. Greater levels of collagen mean greater progress towards remodeling. And in both figures, you can see much higher levels of collagen in the wild type wound as compared to the Granzyme B knockout. Decorin is a protein that regulates collagen fibrinogenesis, spacing, organization, and tight bundle formation. Interestingly, in the present study, we found higher levels of decorin in the Granzyme B knockout wound despite lower levels of collagen. And these interesting results led us to form a new hypothesis. In a pathological setting, Granzyme B cleaves decorin, leading to impaired in collagen formation and eventually to a delayed wound healing time. This phenomenon has been observed in several models, such as skin aging, abdominal aortic aneurysm, and the chronic wounds. However, granzyme B seems to have positive implications in an acute wound model, as we observed in our most recent experiments. In an attempt to explain this occurrence, we looked at the literature, which reveals that granzyme-mediated decorin degradation releases active transforming growth factor, or TGF-beta. The release of active TGF-beta could explain the accelerated wound closure and tissue remodeling that we observed in our most recent experiments. I would like to thank the following people, especially Dr. Granville for letting me into the lab, Dr. Shin for all of his hard work with me, and of course Dr. Williams for all these opportunities. Thank you. Are there any questions?
I have one. This is probably a little off topic, but it ties in with your wound healing. So in your granzyme B knockout mice, did you see keloid formation? Because keloids form because of overproduction of collagen 1 and 3.
Yeah, that's something we definitely looked at. So it's not something that we observed, but it's something that we hypothesized that granzyme B presence kind of could increase the likelihood of keloids happening or other hypertrophic scarring or something like that. Yeah. Yes?
In sort of a chronic wound healing process, and you have granzyme B is releasing the TGF-beta. Do you think TGF-beta in that constant release can restore, decrease in the healing process?
I'm sorry, I didn't hear the second part of the question.
So my question is, I'm thinking about TGF-beta and its roles, and in an acute sense, it makes sense that TGF-beta is a growth factor being releasing, helping the wound close.
Yeah.
But in the chronic sense, too much TGF-beta release, I believe that's the reason why in chronic wound healing sense, you're not getting closure of the wound.
Well, the main difference that we find between the chronic wounds and the acute wounds is that in chronic wounds, granzyme B is super upregulated, and actually the mechanism by which granzyme B is produced isn't really understood, so we don't know why that happens. It could be that excessive TGF-beta could cause even more granzyme B. It's kind of a rolling cycle, and that's how it happens, but we're not exactly sure how all of that granzyme B even builds up in the first place.